I'm Alan Whitmore, and I'm the Chief Scientific Officer of E-therapeutics. Well, it's computing the future of medicine, and I suppose what I mean by that is at every stage of the things that we do, bringing computation to bear on the problems so that we make better decisions. So if you think about what we do, we are trying to make choices about targets, protein targets primarily, that are going to affect biology in a positive way. Then we have to evaluate those ideas in the context of everything else that matters when you're making a drug. Can you find the patient population? What's the commercial environment you're going to be deploying this drug into? What other drugs are going to be used in that disease? Are you going to be able to design a clinical trial that doesn't take years? All of those kinds of things are important. And then after that, when you've made the decision that looks good, you're going to have to design the drug. And then you're going to have to design the experiments that you use to prove that the drug works, and then you've got to get that drug ready for clinic. And at all those stages, you can bring computation to bear. So you can bring computation to bear on the target ID problem, on the target indication evaluation problem, on the drug design problem, on the experimental design problem, and ultimately on the placing things in the clinical context problem. So I think that's the mission statement of the company, to use computation in all of those places to make good decisions. Because if you make a bad decision early on, you magnify the problem down the line. So HepNet is our computational biology platform, and by platform I mean it is a collection of data, so experimental data, information drawn from all sorts of different sources, patient information, papers, publications, patents, and a set of computational tools, because we talked about computation already. And those computational tools are the tools that we bring to bear to get insights from that data. So HepNet is basically computational tools and information, all focused on biology in which liver cells play a part, because liver cells are the cell types that we deliver to. That's what our drug delivery technology is able to do. Hepatocytes are the primary cell type in the liver. There are other cell types in the liver, but about 90% of the liver is made up of hepatocytes. And they are very important cells in the body. They're central in so many different things, probably 500 different functions at least, maybe even more. But some of the functions that might make sense to you are they make all of the proteins that help your blood to clot. They make proteins that are involved in your immune system. They regulate your metabolism, so they respond to insulin, and they are part of the multi-organ system that regulates your blood glucose, so they've got many different functions. Those are just some examples. Well, two reasons. I mean, the most important reason, I think, is that the liver is a really central part of so many aspects of physiology, that if you can manipulate liver biology, you've got quite a good chance of manipulating a lot of different diseases. So focusing on the liver does not tie you to liver diseases. I think it's a very important point to make. The liver makes proteins that are trafficked to the eye and to the brain and to the heart and to the kidneys and all over the place. The second reason is that there is a very important piece of biology that allows you to deliver very specifically things to hepatocytes, to the liver cells. So there's this sugar called GalNAc. It doesn't really matter what the structure is, but there are receptors that recognize this sugar on the surface of hepatocytes, and they can internalize that sugar, and they can basically, if you tie that sugar to something you want delivered to a hepatocyte, you've basically got a postal address. So if you write a letter you want delivered to a hepatocyte, GalNAc is the postal address. You glue that on, then any drug that you make that's got GalNAc on it will be delivered to liver cells and specifically to hepatocytes. It doesn't go anywhere else. So not only have you got the specificity of RNAi, which I guess we'll talk about in a bit, but you've got the specificity of delivery there. And that is unique. There aren't any other delivery systems that I know of that are able to target a cell type with such specificity as GalNAc can. Now in the future, it would be lovely to find equivalents in other cell types, but at the moment, That's the delivery problem to hepatocytes is solved, and I don't think you can say that for any other cell type. So Galomic is another one of these things that's called a platform, and what we mean by a platform there is a drug design and drug delivery platform. So Galomic is the name for our drug design, and our drug design is the way in which we attach that GalNAc postal address sugar to our drugs, and then the way that we design our drugs. It's the special chemistry that we use to make sure that when we design a drug, it isn't broken down by the body, that it gives you good knockdown of the target, and it gives you good duration. Galomic is basically the chemistry side of the platform, and it's a bit of a moot point whether the computational techniques that we use to do the drug design should live under that umbrella of Galomic as a platform, or whether they should be part of HepNet. At the moment, they've become part of HepNet, but it's all part of the same thing because the two platforms belong together. So DNA is the blueprint to make a body, and every cell in your body has got a copy of that blueprint in it. And DNA contains genes, which are sequences that code for proteins. And your DNA lives in the nucleus of the cell, and the proteins that are basically the micro machines of your body are made outside of the nucleus, and so that information has to get from the nucleus to the places where the proteins are going to be made. And that involves an intermediate step. So something comes along, and it doesn't matter what that something is, and it says, I want to translate this gene, I want this gene to be made. And what actually happens then is that gene gets transcribed into another code called RNA, so DNA, gene, into RNA, and you have a sequence in an RNA molecule, and that RNA molecule is the thing that traffics out of the nucleus, goes to the other part of the cell, where it then becomes the template for you to make a protein. And RNAi is an ancient mechanism in cells that has a couple of jobs, and probably in evolutionary terms they ended up as the same job and then got specialized afterwards. But it's a way of controlling how many of those messages actually get translated into proteins. So it's a way of regulating genes at the post-transcriptional stage. And it's also part of an ancient antiviral mechanism. And the antiviral, so viruses make genetic material that doesn't look quite like ours, and in particular they make double-stranded genetic material, and RNAi is particularly able to recognize double-stranded foreign RNA, and that directs the cell then to prevent that from being translated into a protein. And so we cheat. So in effect, siRNA molecules are engineered to look a bit weird and artificial, so that we trigger this machinery to say, hang on a minute, that might be a foreign gene, we need to just switch that off. So we're basically piggybacking on an old immune system. So I guess to summarize, RNA interference is a way in which the cells are able to stop proteins being made from that message. So they basically stop the translation of the RNA into protein. And that's obviously important because you're either wanting to downregulate a disease-causing protein, or you're wanting to downregulate another protein that's going to have a beneficial effect in the context of a disease. Obviously, I'm going to play up the advantages, aren't I, being an RNAi company? But yes, I think there are genuine objective advantages, not least of which is that until very recently, RNAi was an approved modality and gene editing wasn't. There have been some gene editing drugs in late-stage clinical development, and one has been approved. But gene editing does something very different from RNAi. As you can imagine from what I've told you, RNAi will downregulate a protein for as long as there is an siRNA present. But when you stop dosing that drug, the effect gradually wears off and you go back to normal. And that's very important if you have a drug that you need to reverse, or if you don't want to treat for a long time. Gene editing is effectively making a permanent change to the blueprint of your DNA, and that's a much bigger deal. The other thing that's important, I think, is RNAi is a much better known modality, particularly siRNA is very proven technology now, multiple approved drugs, and it's very safe. It doesn't trigger immune responses. in nearly the same way that the vehicles that are used to develop to deliver gene editing do, and that's another big advantage. And the other problem I think with gene editing is it is permanent, but the effects do eventually wear off, and then the real question is, can you then redose? And the evidence is that might be tricky too. So I think lots of advantages at the moment for focusing on RNAi. That's not to say in the future that there aren't them, and there might not be other technologies, but at the moment it's a very good bet. I think, well, so what differentiates us from a lot of biotech companies is actually having access to siRNA in the first place. It's not a straightforward modality to get into. You need a lot of expertise. It's quite a complex IP landscape you have to navigate, and just having the ability to have freedom to operate in that, I think, is an advantage. So that sets us apart from a lot of biotech companies. I think what sets us apart from other siRNA companies is that we have in the same company, and sometimes even in the same head, the computational biology and AI and mathematical and medical experience, as well as the siRNA experience. So we can bring tech and bio together under one roof instead of having to do it via collaborations, which they can work, but they're much more difficult to manage. So I think that's the big differentiator, that we've got that tech and bio together, that we're able to, and also that I think we're big enthusiasts for AI, that we're very aware of all of the problems of AI. Everybody will worry about, particularly things like GPT-4 hallucinating and all the rest of it. Well, we know that. We also know that there are very smart people who are able to find ways around that, to mitigate those problems and to leverage AI in a way that really gives us an advantage. So that combination of AI, computation, and RNAi all under one roof, I think, is our competitive advantage. That's a very good question. So I think how other people use it quite typically is that they'll buy a subscription to ChatGPT Enterprise, and that will be them using AI in a business. That's not fair for everybody, but I think a lot of people, that's the way that they can get into AI. I think we've got more expertise in the company than that. And for us, AI is an opportunity to look at every stage of our journey and add a layer of artificial intelligence to support either the computation we're doing, or in a lot of cases the manual work that we're doing. So if you think about what we do as a company, we do target identification, and then we do target indication evaluation. So target identification is we'll take data, we'll mine it for insights, we'll find a biological process, we'll make a prediction about a target. That's not enough in itself. That's maybe 10% of the solution of what you need to do. You then need to evaluate that in the context of the competitive landscape, the clinical landscape, the investor landscape. There are so many considerations that are layered on top of that choice of target indication evaluation. Can you access the patients that you think you want to treat? Are they going to even present to the doctor at the time in the disease where your drug is actually going to be able to do something useful? So all of those considerations, at the moment, they're very manual, and they involve triaging enormous amounts of information, reading it. You need a lot of expertise in one head. I've got some of that, Ali's got some of that, there are other people who've got some of that, but we're not scalable as human beings. We couldn't do 1,000 target indication evaluations intelligently in one go. Well, with AI, you can address that problem. And then after that, you've got to design your drug, and AI has got something to say there, and we can go into some detail in a minute. And then you've got to design your proof of concept experiments, and ultimately you've got to do all of that in the context of the stuff that you learned when you were doing target indication evaluation. What's your clinical trial going to look like? And all of those things, the downstream, they all reflect back to that initial decision. So I think AI will contribute to our ability to do good target ID, because we won't just be mining data using computational tools. We'll also have access to all of that unstructured information that's out in the world. You could read 1 million papers. I can't, but the AI can, and it can actually assemble that information, put it in the context of the other stuff we know, so we have it talking to our systems, and we can make a better decision about whether a target is likely to have a biological effect. In terms of the target indication evaluation, we, instead of reading hundreds of papers, hundreds of patents, looking at 1 million preclinical experiments that other people have done, we can do that, but we can do that much more automatically. And the way the journey is going to work is it won't all be automated overnight. They'll become decision support tools, and then gradually, as we automate more of the process, it'll become more and more and more scalable to the point where ultimately we might be able to look at 1,000 target indication pairs and just say, well, actually, this is the one that suits this biotech company the best, because you can do your trial in healthy individuals. It'll only cost you this. You'll get a readout in 6 weeks as opposed to 5 years for a cardiovascular outcome trial. All of those things are effectively trying to get you to make, and I've said this before, but the AI is helping you, empowering you to make good decisions as early as possible so that you can walk that critical path and increase your probability of success. So just to come back to that whole concept of AI, it's a buzzword at the moment, but it's been around for a long time, and we've certainly been using what's called AI for a long time. It's a form of machine learning, a form of statistical pattern recognition at its core, and we've been using it for drug design for a long time, and we've been using it for data mining for a long time, and I think that's something you shouldn't forget, that we've got quite a long history in AI before the latest changes that have happened. And what happened a couple of years ago was something that we'd been monitoring for a while, which was this what's called transformer technology. So these things that have become the basis for these large language models that are able to process huge amounts of text. And we were following that field for a while and thinking that the state of the art wasn't quite good enough to do the things that we would need to do. And then something happened around about a year ago that both improved the performance of those kinds of technologies, but also, I think, as importantly, they figured out a way of connecting those to the rest of the world, so that all of a sudden you could have an artificial intelligence that was connected to the internet, that could plug into other things like databases, like maths engines, all of those kinds of things. And I think all of those disparate tools that we were using individually, all of a sudden we saw a way to integrate those. So AI has been a journey, and it will continue to be a journey.